So now we're going to move on, and we're going to welcome a new face to Badger Crop Connect today. She is not new to Wisconsin agriculture, but she is new to extension. So Natasha Rayne, my name twin, and I'm now not the only Natasha in extension. Natasha Rayne comes to us from UW River Falls, and she is now a UW-Madison Department of Soil Science assistant professor and extension specialist focused on soil fertility. And so she has a lot of insights to share with us, to talk about where she comes from, where she's going with things, and how we can hopefully put some pieces together for better nutrient use in our crops. So, Natasha, you can go ahead and share your screen and take it away.
Thank you. Thank you, Natasha. Thank you for that. Sorry about that, folks. Yeah, my name is Natasha Rayne. I joined the faculty here at UW-Madison in January. My presentation today is a little bit different in that I was asked to introduce myself and talk a little bit about my research and research vision, if you will. So a little bit about myself. As I said, joined the faculty recently here. Before this, I was at UW River Falls. I was there for 9 years in this same field, working in soil fertility. And before that, so I did my graduate training at Oklahoma State University, not to be confused with University of Oklahoma, two completely different things. Anyways, Oklahoma State University did both my master's and PhD there. And even before that, I was in Suriname. And the educator in me wants to give a quiz right now and ask how many of you know where Suriname is. Suriname is on the northeast coast of South America. We're bordered by Brazil, Guyana, French Guyana. Suriname is also considered part of the Caribbean, right? So go figure, right? We're in South America, considered part of the Caribbean, and the language spoken there is Dutch, right? So if you're confused, I understand. Yes, my first language is indeed Dutch. So that's my educational background. Did my agronomy degree in agronomy at the Anton de Kom University in Suriname. And yeah, and worked for a couple of years in different fields in agriculture and so on. But talking a little bit more about my professional experience. So a funny thing is that, we all know probably that life is not a straight path, right? Did my degree in agronomy, undergrad degree in agronomy. And my first job out of undergrad was in a food processing company. And you wonder, how do I go from agronomy to food science? Well, my bachelor's degree had a thesis component, right? A thesis requirement. And just like what you would encounter in grad school, right? The professor that has money or has resources takes you on as a student, or you request that you be part of their lab. So that is how I ended up with a food scientist as a supervisor for my thesis project. And that opened up a lot of other opportunities out of undergrad, right? Like, so my first job, as I said, was at a food processing company as a quality manager. Right. It also opened another door in that I got certified as a educator in food safety. The government in Suriname was big around that time when it comes to food safety for, these canning companies and food processing companies, right? And I was certified as an instructor to be able to train these employees, if you will. So so that was that. Moving on from there, I kind of missed that connection with agronomy, missed that connection with the field, right? My second job out of school was as a farm manager at a banana company. Suriname is a big exporter of banana. Rice would be the #1 crop. but banana is a close second to that. We have approximately, this company I worked for had approximately, I would say 5,000 acres, if I do the math right in my head, from hectares to acres, you know, and I worked with, I was one of the many farm managers that they have. I think one of 6 farm managers. And underneath me, I had like 120 field and packing station workers, 3 supervisors, and then 6 foremen. So it was, you know, the farm managers, 6 foremen, 3 supervisors, foremen, and then the people who did the real hard work, right? So that was that. Well, and moving on from that job, because it was hard work. It was hard work, it was day and night, it was weekends sometimes. Lasted approximately a year there because yeah, health reasons, right? And also, yeah, just the pressure, I guess, yeah, and health primarily. Moved on to a job that I really enjoyed, which was basically my dream job. This was as a project coordinator. That was the official title, but it was really an extension, because my job was, I worked for an NGO. This was an NGO that hired me as a project coordinator, and they were basically helping subsistence farmers, right, with small loans, and you helping them run their farm as a business, right? And our job as agronomists was to basically provide that technical knowledge, right, the technical side of things, field side of things. So I was in charge of the extension officers. It was a small team. It was a small team, but still, you know, I was in charge of that, leading that effort, organizing training sessions, et cetera, et cetera. So I was responsible for that. You know, and then I spent a couple of years, right, in all these different management positions, going from quality management to farm management to now extension and project management, if you will, right? And you feel clearly that, okay, well, I've reached the peak. Okay, what is next, right? Where do I grow to next? And that is when I started pursuing graduate school. So straight out of grad school, then fast forward, right? I spent the last 9+ years in academia, teaching, doing research, and now full on extension. Moving on from there, please. So now I want to talk a little bit about my past, present, and future research. Please know that I'm not going to get too technical in detail, talk about each one of these projects or research topics, but rather just briefly talk about just some big themes here. Yes, please move on. Thank you. All right, so my training is in nitrogen management, right? Nitrogen management, nitrogen use efficiency, right? And I come from a program, you know, during grad school where I worked a lot with crop sensors. Okay, now these crop sensors basically rely on equations, algorithms that have been developed over time, over a long period of time, with a lot of data, right, to establish a relationship between these vegetation indices, such as NDVI, and yield, right? So they look at the relationship between NDVI and yield, and then make calculations about, okay, what should be the nitrogen rate to apply. Now, I went a different route I started asking different questions using these vegetation indices. I wanted to look at things like, okay, the relationship between NDVI and grain protein, right? I worked with winter wheat a lot. So I was interested in that relationship. Why? Because we know that nitrogen rates also affect grain protein, right? So a different angle, but still working with these sensors to kind of answer a couple of different questions. I also wanted to see, hey, can we predict how efficient the uptake of nitrogen is by the crop? So predicting nitrogen use efficiency using NDVI, again, to make informed decisions about nitrogen to apply. and so on, and predicting mid-season response to nitrogen, because that's really what these sensors do, right? They can only help us mid-season, not so much determine pre-plant nitrogen rates, right? They can't do that. They usually are used to do mid-season, in-season nitrogen application, right? They help us, they inform us about that. So I did some work with that. This was, that's me, a publication by me and me and a group, of course, and we looked at just different models, different predictor models for NDVI and yield, NDVI and protein, NDVI and nitrogen use efficiency. And what we concluded from these studies, which is not one study, it was many different studies that we looked at, that the NDVI, which is vegetation index, again, in and of itself is not enough, right, to help us predict, let's say, the response to nitrogen, nitrogen use efficiency. We needed more than that, and that's where we needed to look at different environmental variables, right, that we could incorporate in these models. Moving on. I touched on this already. We can just keep moving. Another thing that I did on the side, and I really enjoyed this little project. It wasn't part of my thesis or anything, a main assignment. I came together with a group of computer scientists, and what we did is that we really dug into data on nitrogen loss in the form of ammonia from surface applied urea, right? And we really put all these data together and developed this online calculator. And I say I really enjoyed this because I think it's a helpful tool. And really that's where my heart is, right? Basically developing simple tools that can help farmers in their decision making about nitrogen management. We can move on. All right, so then I moved to Wisconsin, and now I began asking different questions, different types of questions using these crop sensors, right? For example, I remember my first summer in Wisconsin having to look up at the corn. Right now in Oklahoma, we simply didn't have enough moisture in the soil to grow tall corn, right? So that was a revelation for me, walking into a field and having to look up at corn growing. So I began asking different questions about when it comes to these crop sensors, right? Can we predict things like, I told you that one of the projects that I looked at was grain protein. Okay, well, can we basically apply that to predicting, let's say, quality parameters in corn for silage, right? So I started looking at crude protein, for example. Can we use these sensors, and how does it relate back to nitrogen rates, right? And can these vegetation indices help us to predict the response to additional nitrogen in corn and so on? This is the result, or at least one of the graphs that came from the study looking at NDVI and crude protein, and we were not really successful in predicting crude protein based on the NDVI. So it's just 1 year of data here represented, but is it worth looking or pursuing this into the future? Maybe, right? At this point, I would say it was inconclusive, right? So yeah, we can move on. All right, I continued work with looking at the NDVI as a predictor of yield, right, and as a predictor of the response to nitrogen. And we found in this particular study done at Lake Farms, right, that V8 was the right growth stage to go in, and or the best growth stage in when it comes to the prediction of yield and the response to nitrogen. We can keep moving. Then I did more of the traditional, also more of the traditional nitrogen rate studies, such as a split application and looking how it affects yield and how it affects nitrogen recovery, nitrogen use efficiency, and that sort of thing, right? This particular study, or these studies, were done on stilled loams, high yield potential. We didn't see any significant differences in terms of the growth rate. We looked at things like the growth rate, yield. I think we did protein analyses too on these samples, or in these different treatments. We can move on. Yeah, this is another example. No matter how we did the split 4 ways, 3 ways, 2 ways with different rates, we did not see a significant difference in yield between treatments. Another aspect of these split applications that we looked at was also nitrate, right? Nitrate in the soil. I think these samples were taken at 2 feet deep. And what you see is kind of what we expected. If you do a single application, you see a quicker drop in that nitrate, soil nitrate, as a function of time compared to when you do multiple applications, right? So that's that. But then you're in Wisconsin, so all of a sudden your focus also changes, and I'm talking about my focus changing, right? Where the focus, one of the crops that we grow in Wisconsin is manure, of course, right? So anyway, so manure. I needed to switch my focus to manure, and I began looking into manure as a some questions that I was interested in was, how does it affect soil health, right? I know that soil health is a big term, right? Soil health is defined in different ways, and I don't know that we have completely figured out all the parameters that go in soil health, but I produced papers that looked at physical parameters, soil parameters, chemical parameters, biological parameters, and so on and so forth. We can move on. A couple of questions that I asked with manure and am still asking today, or looking at manure and the nitrogen credits from manure. So we wanted to see the availability of nitrogen from manure. No surprises here. If we compared manure applications to, say, urea, we saw a quicker release of that nitrogen, nitrogen from urea in comparison to manure, because we know that manure needs some time in order for that nitrogen to mineralize and be released into the soil. Speaking of soil health, I told you that I was interested in that soil health component or soil health aspect of manure, right? We looked at things like CO2 burst, for example. Interestingly enough, as you can see from these data real quick, without, again, getting too technical, the higher manure rates gave us also the higher CO2 burst. No surprises there, but we also see that we collected these samples before planting, and then prior to harvest, you don't see any decline in that CO2 burst with the higher rates of manure that were applied, right? Whereas if you look at urea applied at 150 pounds of N per acre, then you see a quick drop, or not a quick drop, but you see a quite significant drop in the CO2 flux, if you will. We can move on. All right, so a big component of my research currently here, and these questions that I have been asking for a while, nitrogen recovery, digestate, anaerobic digestate, right? We see an increased interest in anaerobic digestate, liquid solid separation. And I think it would be of benefit to us, and that's definitely a need for us here in Wisconsin to start looking at these products and looking at the nitrogen recovery from these products. And I made an effort in starting some of this work. This is a study that we did in the area of River Falls, where we looked at different manure products and looked at the nitrogen recovery from them. Interestingly enough, the digestate applied at single rate. We did a single rate, we did a double rate, 2 times the recommended nitrogen rate. We see that the one applied at a single rate of nitrogen gave us the highest percentage recovery. But then again, you know, nitrogen recovery was all over the place, 2 to 52%, right? So yeah, so that's that. A project that I'm still involved with, from River Falls, out of River Falls, looks at, and this is definitely also one of my interests, or a question that I have as it pertains to manure and nitrogen credits from manure, is definitely understanding application methods and nitrogen availability. So my task, or my role on this project, by the way, let me give a little background on this project. So this is looking at manure application technology, right? It's basically a frame that that comes in, or that has 3 different configurations, right? 3 different configurations. One is a sweep injector configuration, a disc injector configuration, and a vertical tillage configuration. And what we'll do is basically just look at different application methods, these different application methods, and then mixing the soil in, and tillage and so on, because it's really a tillage implement, right? And looking at the nitrogen distribution as a function of that manure that is applied, right? So the manure is applied and then worked into the soil in different ways, right? 3 different ways. And what we'll look at, or my role, is to look at the nitrogen distribution as a function of that mixing, and as a function of that incorporation, and then nitrogen recovery, right? Because again, the big question here is that, how does it perform, or how does the manure do when it comes to the availability of nitrogen? So that's that. So what is, you're hearing all this, right? All these different projects and ideas and background and whatnot. And you're like, okay, Natasha, what is your research vision? Well, I'm glad you asked, because my research vision is to really integrate some of these canopy sensors and remote sensing into our nitrogen management. Basically finding ways to, can we refine our current nitrogen recommendations using some of these sensors, right? And then when it comes to manure and legume credits, I should say, with manure, I think there's an opportunity to expand on the work that has been done already. And as I mentioned before, just looking at some of these other products, right? And answering questions like, for example, a question that came up the other day is, how does dry matter percentage affect nitrogen availability? Here in Wisconsin, right, we have this separation between 11% dry matter and then 11 to 20% dry matter. And the question really is, does it matter or not, right? So does it matter, right? Whether it's 11% or 20% dry matter and so on. That's a simple question, a big question that we want to help answer when it comes to nitrogen availability from manure. And as I mentioned on that last slide with the project out of River Falls, I also have an interest in looking at application methods and timing and nitrogen availability. All right, I think that is it when it comes to my story, my background, and my vision. If there's any questions, I'll be happy to answer them. Thank you.